Hello, my name is Spencer Fisher, and this is my video for this week's lab. Now, in front of me, I have all the things that was given to us for this lab, and then I have my draw out of everything for the mitosis and the meiosis breakdowns. Now, when it comes to the first questions that we had to answer, mitosis and meiosis are important just because they both involve the growth, cell growth, for healthy cell growth and for sexual reproduction cell growth. And the mitosis, it takes place all over the body, head to toe, all over the body. Where meiosis is just in the cell, the sexual reproduction cells, so sperm cells and the egg cells for women. So I'm going to start with the mitosis. I'm going to adjust this so you can see the breakdown. Mitosis. Now it starts with the intraphase, which is the G1, the cell growth. That's where the cells grow larger, and then you have the S phase where the DNA replication and duplication happens, and then you have the G2, which is the preparation for nuclear division. And so then it goes to the prophase, and the prophase is just simply the DNA condensing into the chromosomes. And so that's what would happen here, to where they attach themselves to the cooled sister chromatids to hold their structure together. Then it would go to a prometaphase, to where it comes in and connects in the centers to pull them apart, which goes to the metaphase, where the sister chromatids migrate toward the metaphase, which is what you see right here. They would line up as such, and they grow and they link together, attaching, and they attach to each pair of the sister chromatids in the middle, like I was saying. In the anaphase, they're attached to it and they're shortened, causing the sister chromatids to separate and move toward the opposite poles of the parent cells. And I have two quarters that represent the two parent cells on the outside, just to help me memorize it. So what would happen is they would separate and get pulled this way, individually, going away from each other, and it would line up like such, to where it is broken down like this right here. Then that would go to the telophase, where one set, this is the telophase, where one set of the chromosomes arrive at each pole. And this is where the new nuclear envelope begins to form. They're uncooled, and their new nucleus is formed here, which will turn it, and they'll all combine together. And this is the replication of the new nucleus is there. And that's the final stage. That's where the division into the two daughter cells is complete, like such. And that's it for the mitosis phase. So then we would go to the meiosis 1. The meiosis 1, and it starts with the prophase 1. So, and again, that would go back to the sister cells, like such, for meiosis 1. Now, in meiosis 1, this is where they're cooled to themselves, the sister chromatids, to hold their structure, just like in the mitosis phase. Now, this is also where crossover may occur at this point. And that just means that the chromosomes may exchange and recombine regions. And I'll demonstrate that. I'll come back and demonstrate what that would look like at the end of the video. That way I can, cover that as well. So we pair them together with their sister. Like such. And again, you have it to where it breaks down. You might have two over here and two over here. And then that's that. And the crossover, it just creates a unique genetic fingerprint. And like I said, I'll go more in depth with that when I get back to it. And you go to the metaphase one, which is where the chromosomes migrate toward the metaphase plate. That's where the microtubules, they grow, and what did happen is they'll realign, and they'll be connected from the parenting side to the center of it. The centrals, I think is how it's pronounced off the top of my head. And they'll either link together or attach to each pair of the chromosomes at their center. And then the antiphase is when they'll get brought to the parenting side. It's attached to the chromosomes, shortening, pulling the chromosomes apart and moving them to the opposite poles, like you see here. Let me try to align this better into the camera. All right, so like such, they'll be off to the sides like that. And then that would take you to the telophase 1, where you will have the new nucleus, and that would be a nucleus, and that'll be a nucleus. And the telophase is just where one set of the chromosomes arrive at each pole, and a new nuclear envelope begin to form. And that's the new nucleus is formed right there, like such. And then you could have the meiosis 2, which is where you would bring it like this. And the meiosis 2, now the meiosis 2 is more of a, the prophase is where it starts again with meiosis 2. And so it's like that, and that's where the new spindle fibers form for as the nucleus breakdown again. And it would kind of, the prometaphase would attach from the parenting cells or the parenting poles, and it would take you into the metaphase to where you'll see them line up more so like that, and to be attached like such to the center, represented by these beads. That would go to the anaphase, where the sister chromatids are separated. So you would see them being separated apart and getting pulled to the opposite poles, like such. They would separate and get pulled away. That way there's one on each side of the poles, like such. They're there and they're there. All right, and then that goes to the telophase, where 23 chromatids arrive at each pole. At that time, a new nucleus forms around them. So they would all connect once they got there. All the chromatids would connect, and then a new nucleus would form around that, and that would be your new nucleus. That's for meiosis 2. Now back to what I was saying about the what the crossover would occur as in meiosis 1. So you would have something that looked a little more like this. So you'd have the two, like you might have these two as sister that combine together. And you would have these that changed over. They're not sistering, they're not the same, but they switched, and they attach to each other like such, which would cause these to attach to each other as such. And then what would happen there with the metaphase is, once they get attached, once they get grabbed and they get pulled, it would look more like that, to where there's opposite, the crossover like that. And that's all the crossover is. It would still go the same, just with the crossover, with all that. I hope this video covers everything. That's all I got for you.